Good morning, teacher and classmate. We are the Anonims Anemones team, and today we will talk to you about a biomonitoring case. And this case is about the biomonitoring of the Western European rivers Rhine and Meuse. First, I would like to provide some context on the topic. Biomonitoring is based on the study and measurement of biomarkers in native species, as it allows for the assessment and identification of harmful effects of chemicals in the environment. Biomonitoring presents a wide range of advantages over environmental monitoring and has been considered to be a valuable tool for both ecological and human health surveillance. Cautious interpretation of data is one of the most sensitive issues on this matter. Still, the development and application of biomonitoring lead to improved knowledge of the effects of chemicals on living organisms and at the same time to increase the protection of public and health and environment. The Rhine River, a vital waterway in Europe, holds significant economic importance as it facilitates trade and transportation through Germany, France, and Netherlands. Serving as a key commercial route, the Rhine plays a crucial role in regional economies, fostering trade, industry, and economic development along its bank. As you can see, there are two maps. In the left map, only the main river is marked, while in the right map, the rivers stemming from the main river are shown. Many hundreds of animal and plant species live in the innumerable different habitats along the Rhine and its tributaries. Each species is adapted to the particular ecological characteristic of its habitat, to water temperature, velocity of current, oxygen, and nutrient content. The presence of a particular species permits conclusions regarding the ecological state and of the habitat. Therefore, some fish typical of a specific habitats on the other water organisms serves as indicators for the ecological state. Plankton and waterfowl also play an important role. The last biological inventory showed that many species of fish as well as invertebrate fauna, including snails, insect larvae, and mussels that were regarded as extinct in the Rhine have returned. In spite of this success, today's aquatic communities in the Rhine are far from having reached a stable balance.
Zebra mussel is scientifically known as Drosenia polymorpha. Zebra mussels are invasive freshwater mollusks native to the Black Sea region of Eurasia. They have been introduced to various water bodies around the world, primarily through ballast water discharge from ships. Zebra mussels are considered bioindicators. This meaning the presence or absence can be used to assess the health of an ecosystem. They are highly sensitive to environmental change and pollution levels. The presence of several mussels in a water body may indicate good water quality, as they thrive in clean, well oxygenated waters. Conversely, their absence or decline may suggest environmental issues or pollutions. Zebra mussel is scientifically known as Drosenia polymorpha. Zebra mussels are invasive freshwater mollusks native to the Black Sea region of Eurasia. They have been introduced to various water bodies around the world, primarily through ballast water discharged from ships. Several mussels are considered bioindicators. This meaning now talking about ecological impact. Several mussels are filter feeders. This meaning they can extract particles from the water to feed on. This can improve water clarity by removing suspended particles. It can also have negative consequences. The extensive filtering capacity of several mussels can lead to the depletion of phytoplankton, the primary producers in aquatic ecosystems. This may impact other organisms in the food chain that rely on phytoplankton as a food source. And now, the negative ecological impact of several mussels can have detrimental effects on native ecosystems. The rapid reproduction and colonization can lead to the displacement of native species, alteration of nutrient cycling, and change in water quality. The depletion of phytoplankton by several mussels may affect fish population and other aquatic organisms. Now, talk about economic consequences. Why not the commercially exploited as a food source? Several mussels can cause economical problems. They can attach to hard surfaces such as pipes, water intake structures, and bone holes, leading to clogging and increased maintenance costs. The economical impact of managing and mitigating the effects of several mussels in invade water body can be substantial.
The biomarker used in this biomonitoring case was the concentration of heavy metals present in several mussels, which are cadmium, zinc, and lead, used to analyze the contamination of the Rhine River water by these heavy metals, which has been threatening the life of this ecosystem. Chemical nature. Cadmium, it is a silvery white metal, very shiny, very ductile, and malleable. Its orange vapors crystallize upon cooling into regular octahedral shapes. It is quite reactive against oxygen and acid, but not against alkalis. Its chemical structure is a compact hexagonal packaging with a molecular weight of 112.4 grams over mole. On other hand, zinc is a malleable, ductile, and gray metal. Zinc is one of the least common elements and is estimated that it is part of the Earth's crust by 0.0005 to 0.02%. It occupies 25th place in order of abundance among the elements, and its molecular weight is 65.4 grams over mole. And finally, lead is a heavy metal bluish in color which tarnishes to acquire a dull gray color. It is flexible, inelastic, and melts easily at 327.4 degrees Celsius. It is relatively resistant to attack by sulfuric acid and hydrophobic acid, but dissolves slowly in nitric acid. It has an amphoteric behavior, that is, it reacts with acid forming acid salts, but also with bases forming metallic salts and oxides, with a molecular weight of 207.2 grams over mole. Biological role in several muscle. These organisms has the ability to withstand a high metal concentration factor without suffering mortality. Dreissena polymorpha is attached to hard substrates above the sediment by It bises threads and, as a filter feeding organisms, lives in close associations with the water phase. Concentrations of heavy metals in these muscles may reflect the ability of heavy metals for filtering feeding organisms. Furthermore, the polymorpha is the main source of food for benthivorous fish and for diving dogs. Bioaccumulation of contaminations by the polymorpha, therefore, is an indication of potential harm for these predators. Identification and quantification technique. The identification and quantification of heavy metals in several muscles were carried out using a digestion method with ultranitric acid and hydrogen peroxide to later be analyzed by atomic spectrophotometry.
Biomonitoring can provide valuable information about the state of ecosystems, but it also has ecological and economic consequences, both positive and negative. Some of these are in ecological impact. Biomonitoring helps detect changes in ecosystems over time, providing valuable information on environmental health. By studying the presence and behavior of indicator species, biomonitoring can offer insights into the overall biodiversity and ecological balance of an area. S- um, Negative ecological impacts can include the process of collecting organisms for biomonitoring can potentially disturb habitats and ecosystems, affecting the very environments being studied. If certain species are constantly used as indicators, it may create selective pressures on those species, influencing their populations and potentially altering the natural balance. Also, the use of non-native species for biomonitoring can sometimes lead to the unintentional introduction of invasive species, which may disrupt local ecosystems. Some economic consequences include biomonitoring can help identify environmental problems early on, potentially preventing more costly consequences in the future. However, collecting, analyzing, and interpreting data from biomonitoring studies can be resource intensive, requiring funding and expertise. Negative impacts on ecosystems can have economic consequences as ecosystems provide various services like water purification, pollination, and climate regulation, which could be compromised. If biomonitoring identifies environmental issues, it may lead to the implementation of regulatory measures to address these problems, which can result in economic costs for industries and businesses. While biomonitoring is a valuable tool for assessing environmental health, it is crucial to consider both positive and negative ecological impacts, as well as the economic consequences associated with its implementation. Balancing the benefits and potential drawbacks is essential to ensure effective environmental management and conservation efforts.
By monitoring, studies can be either active (APM) or passive (PPM) to study trends into bioavailability of heavy metals in the rivers Rhine and Meuse from 1976 to 1988, and to locate and discharge. Both APM and PPM were carried out with the polymorpha. Such studies were always done from October to December. The bioavailability of cadmium in the river Rhine and Lovit has decreased during the last decade. We can see in the figure 1 this information. While the cadmium concentration in muscle exposed in the river Meuse at Hden ha- has increased. Um, Copper concentration in muscle from both rivers have not changed, like we can see in figure 1. Now, lake. Isilmere, including Lake Markham. It is a suitable reference area for ABM studies. Seasonal variation in the heavy metal concentration in the polymorpha take place possibly caused both by change in the bioavailability of contaminants and by the annual growth and reproductive cycle of the animals. In the long term, EBM studies one should take care that animals are collected at the same location and in the same season. And in the figure 2, we can see the average concentration of different heavy metals in Dresemena polymorpha from four locations for different seasons.